Hello and welcome to the sixth video in the topic of cell biology, and this video will be about cell division, which is arguably the most important part that you should remember from cell biology because it is necessary in the subsequent topics of IB biology, which are yet to come. So to talk about cell division, I'll first talk about the different kinds and forms that the DNA is coded in and how it is transported and transformed. Next up, I'll talk about interphase, or before that, I'll briefly talk about the cell cycle as a whole, and then move on to interphase, mitosis, also including cytokinesis, and lastly, cancer and the formation and creation of cancer, which is an example of faulty cell division. So yes, moving on to the DNA, we have the different kind of forms, and before I can talk about the different phases in mitosis, I should probably talk about the different kinds of DNA. So we have chromatin, which is DNA usually loosely packed and unraveled in all non-dividing cells. So chromatin is the state of the DNA throughout interphase. Then we have a chromosome, which you can see in the picture. That is DNA temporarily packaged into tightly wound and condensed versions prior to division, and it's easily segregated but inaccessible to transcription, and that is also what you can see because of karyotypes when you look at the genetic makeup of a person. We lastly have chromatids during the S phase of the interphase, where sister chromatids separate for mitosis into chromosomes, and you can see that these X-shaped chromosomes have two chromatids. Moving on to the cell cycle. The cell cycle is the thing that new cells are needed to replace cells that died or to allow organisms to grow, and cell division is essential but must be controlled. So we have the M phase, or the mitosis, including prophase, metaphase, anaphase, telophase, and cytokinesis. Then we have the rest of that is the interphase, as you can see. So we have the G1 phase, the growth and normal metabolic roles during the first growth phase. We also have the synthesis, or the S phase, that includes DNA replication, which will be a topic in molecular biology coming soon. And then lastly, we have the G2 phase and the growth and preparation for division. And I'll be talking about all of those in detail. The key processes are that DNA replication and organelle duplication and cell growth, transcription, translation, obtain nutrients, and cellular respiration are integral to the cell division. Yes, so the interphase is the largest part of the cell cycle, and it's the active period for metabolic reactions, and the cell performs the pre-programmed tasks. So what it has been differentiated to do for a certain cell type, it will do that throughout the interphase and be briefly interrupted throughout mitosis. And we have the first growth phase, which, as you saw, is there for growth and the normal metabolic roles. It takes place after division. The cell grows, the DNA is transcribed, and the protein is synthesized. Then throughout the S phase, or the synthesis phase, we have the DNA replication in the nucleus of the DNA. In the nucleus, the DNA is made into chromosomes and is then replicated. Then the second growth phase is the growth and preparation for actual division. So there we have rapid cell growth and protein synthesized for cell that prepares for this division. And many proteins are necessary for division, and they need to be synthesized at the ribosomes in the cytoplasm throughout the interphase. And the number of mitochondria increases, so respiratory rate can be rapid to provide energy for division. Moving on to mitosis, which you can see on this graph or picture here. Mitosis is a form of asexual reproduction, and it's the process of nuclear division where the DNA is duplicated and arranged into two separate nuclei. So first we have the early prophase, where the DNA continues condensing into chromosomes and the nuclear envelope, so kind of the membrane around the cell, begins to disintegrate. We move on to the late prophase, where the condensed chromosomes appear as chromatids held at the centromere, so the center part of the chromosomes, and the spindle, the nucleic spindle, no, the mitotic spindle begins to form. The centrioles move apart, which are kind of the poles of the cell. The microtubules form part of the developing spindle. Next up, there is I think here it's set as pro-metaphase, but it can also be called the late prophase, what I just talked about. Then we have the metaphase, which is the part where the microtubules attach to each centromere and move the chromosomes to the equator, which is then the mitotic spindle and the process of depolymerization. The second to last phase is the anaphase, which is the continued contraction of spindle fibers that cause the generation of identical sister chromatids to separate. And once chromatids are separated, they're considered individual chromosomes and move to opposite poles of the cells. And last, we have this telophase, which is once the two chromosome sets arrive, the fibers dissolve and chromosomes decondense, the nuclear membrane reforms, and cytokinesis occurs, splitting the cell. And cytokinesis is the process of cytoplasmic division, where the cell splits into two identical daughter cells. And in animal cells, or centripetal cells, it takes place after the anaphase, where the microtubule filaments form concentric ring around the center, which furrows the cleavage between the cell. And on the other hand, in plant cells, it also takes place after the anaphase, but carbohydrate vesicles form at the equatorial plane and fuse together, throughout which the cell plate forms, which is visible in binary fission. And throughout all of that, the supercoiling of the chromosomes takes place. So we have the normal DNA as you know it in the double helix form, surrounding histone proteins making up nucleosomes. And it then, throughout the mitosis, starts to cylindrically coil to chromatin fibers, where last we have this very condensed version that can be transported and easily separated. In the metaphase we have this chromosome. So that is basically what mitosis is all about. And we also have the aspect of cyclins, which are protein compounds involved in the cell cycle control, ensuring that cells only divide as required. And it is important in detecting and repairing genetic damage and preventing uncontrolled cell division. And cyclins interact with proteins, so-called CDKs, which are so-called cyclin-dependent kinases, and these proteins are regulatory. They form enzymes that direct cells through the cell cycle through phosphorylation. And this phosphorylation is, first the cyclin binds and activates the CDK, so the cyclin-dependent kinases. Then in the second part, the CDK activates target protein to regulate the cell cycle. And lastly, the cyclin is destroyed, allowing for this cycle to occur again. And here we have the cyclin expression cycle throughout the cell cycle. So there is in general, in another diagram, it would show the MPF, the metaphase promoting factor, where the MPF activity is directly correlating to the cyclin concentration, and the MPF activity is always higher than the cyclin concentration. And by the onset of MPF activity, the cyclin concentration rises substantially, and that is also why the mitochondria concentration is higher throughout that part, not only for cell respiration, but also because of cyclins. And we have 4 types of cyclins that take place at different checkpoints. And checkpoints are regulatory points. So for G1 phase, here you can see the growth condition in DNA damage. The G2 phase is the checkpoint for the state of the size and replication, if the replication is faulty. And another checkpoint is at the metaphase, which is when this proper spindle assembly takes place. And the cyclin types are, for one, the G1 cell type, which coordinates cell growth and start of new cell cycle. The G1 or the S phase cyclin starts the DNA replication and promotes centrosome duplication. Solely at the S phase, it induces DNA replication and promotes events of early mitosis. And lastly, at the M phase, the influence formation of spindle alignment of chromatids, which is what you can see in the concentration throughout the different phases in this diagram. Lastly, for mitosis, we have the mitotic index that shows the ratio of number of cells undergoing mitosis and the total number of visible cells. And this is an important prognostic tool for cancer, actually. And the index is calculated by dividing the number of cells in mitosis through the total of number of cells. Moving on, we have the aspect of cancer, which I have mentioned throughout this presentation. And cancer is a disease caused by growth of tumors that are known as cancer. So most normal cells undergo programmed cell death or apoptosis, but cancerous cells have lost their normal control mechanisms and divide uncontrollably, which then creates the tumor. And there are 2 genes involved in creation of cancer, which are the genes that control the cell cycle. We have, for one, the proto-oncogene that start the cell division, and the tumor suppressor genes that switch off cell division. And mutations can make the proto-oncogenes oncogenes that lead to uncontrollable cell division, resulting in cell becoming immortal and non-functional. And when the tumor suppressor genes are mutated, this switches off the stop for cell division, which again creates these immortal and ever duplicating genes. These are affected by mutagens that modify the DNA as physical, chemical, biological agents that damage the DNA and cause mutation through, for example, radiation or carcinogens. For example, when you have smoking cigarettes. You can hear about it more in detail in my video on nicotine. And these carcinogens are agents capable of causing cancer with long-term exposure that accelerate the rate at which dividing cells make errors. And here you can see the process of the creation of cancer, and that is, at first you have the mutation and the formation of a primary tumor, and the primary tumor can either be benign, so it's harmless and doesn't spread and doesn't cause this vascularization, but if it is malignant, it is harmful and it causes this vascularization, which is the formation of capillaries throughout these cell groups, which causes it to spread. And at the process of metastasis, these new capillaries provide route for malignant cells to break away and transfer to other parts of the body and start new cancers. And as you can see here in the detachment and intravasation, this malignant cell transforms to the extravasation, which then is the invasion of a cell group, forming a secondary tumor. And this cycle repeats with the vascularization of the secondary tumor and ends up becoming cancer in the last form. So this was my presentation on cell division, and this was the last topic of cell biology as a whole. Thank you, and if you want to know more about cell biology, check out the playlist. And as always, thank you for watching, and I hope you have a nice day.